What happens? Cells are dying. They are regenerating. From where? These stem cells. So they are needed for, by apoptosis. I think it is already been, Balaram sir has explained, class 1 apoptosis. So definitely in the coming week it will be there. So apoptosis, the cell, we call it programmed cell death. So the different cells are constantly dying, so they are replaced. Okay, and this removal of these dead non-working cells, they are removed. The process of removal is become before 9. This removal, short of removal, is called apoptosis. However, so these cells are replaced by these stem cells present in different organs. Okay, so this type of stem cells are also present in our bone marrow, and their function is not to develop all kind of body cells. Their function is hemopoiesis. Their function is hemopoiesis. What is hemopoiesis? Production of blood cells. Maybe it platelets, maybe it granulocytes, maybe it agranulocytes, maybe it erythrocytes. So all are the process we call hemopoiesis. Okay, so in the bone marrow, there is one kind of stem cells, we call them hemopoietic stem cells. H for hemopoietic, S for stem, C for cells. So what is the abbreviation? HSC. Okay, hemopoietic stem cells. So these, the stem cells present in our bone marrow, what is the name? Hemopoietic stem cells. Okay, now we are coming to definition. The process by which matured RBCs are generated, formed from where? What is the cell? Hemopoietic stem cells. Okay, so the process by which hemopoietic stem cells generate erythrocytes is called erythropoiesis. And now it is clear to you. So we learned what is erythropoiesis. So it is the process by which RBCs are formed from hemopoietic stem cells into RBCs. Okay, erythrocyte. And this process involves three important things. One is development, another is differentiation, modifications in the cells, okay, so, and maturation. We know reticulocytes are immature red cells. So when they are matured, then we get erythrocytes, matured RBC. So maturation is also involved. So what are the three processes we will be involving? It will be involving development, differentiation, and their maturation. Okay, clear? Fine. And starting from the stem cells to erythrocytes, how much time is required? 21 days. Okay? These SSC cells, they produce some progenitor cells. You know what is progeny? Some progenitor cells are produced. Who produce? Stem cells. Okay. This takes production of progenitor cells from stem cells, it requires 17 days. Okay? And from there, these progenitor cells to RBC, it takes 7 days. How much? 7 days. So, from out of these 7 days, 2 days are required for RBC, reticulocyte to RBC. That is, for maturation, it takes 2 days. 5 plus 2. And in total, 21 days. Can you remember this? Will you remember this? Okay? 21. Total 21 days. But in our body, there are certain influencer. No. In my body, there is emergency. I can't take so much time. Every process should be fast. What is the situation? Loss of blood. Because of hemorrhage. Okay, I have been put in high altitude. So in such conditions, body will produce something, they will hasten the process. In that case, time will be shortened. Okay, not 21 days. Particularly the earlier phase, that 17 days, that will be shortened. Okay? But the remaining time, 7 days plus 2 days, 5 days plus 2 days, they will remain same. Only thing, if it is very urgent for me, immature blood cells will be coming into the circulation. Say for example, reticulocyte. Understood? When reticulocyte is coming, when demand for RBC is much more, then only reticulocyte, normally it is 1% of total RBC in circulation. But in such case, it may be increased to 4%, 5%. So whenever you will find my reticulocyte, my patient reticulocyte count is increased, immediately you think that the patient bone marrow is active, very much active. It is trying to produce lot of RBCs. Definitely there is something wrong. There is scarcity of RBCs. Why this scarcity of RBC in the blood? There must be anemia. And if the patient is anemic, the patient must feel hypoxic. Okay, so hypoxia, that because why? Anemia. Why anemia? There is less production of RBC. Why less production? Various factors. Various causes of anemia is there. We'll take one class for this. Okay? So, during this process of erythropoiesis, we'll find, particularly from progenitor cells, these are pro-erythroblast. What are these progenitor cells? Pro-erythro, we'll be discussing. Pro-erythroblast. This pro-erythroblast to RBC formation, mature RBC formation, we'll see 7 important characteristic changes during the process. Number 1, what we will observe? The cells will be dividing very fast. Progenitor cells will be dividing, the number will be increased. That means mitotic activity will be there. And after that, initially there will be more mitosis. Gradually the rate of mitosis will be lessened. And when reticulocyte is formed, they will lose their mitotic activity. Okay, so before their mitotic activity was there, gradually it was diminishing. So this is number 1 observation we will see when we will be studying the different stages. So what is that observation? Their rate of mitosis, number of cells will be increased, rate of mitosis will gradually fall and become absent. Okay. And one more thing, we know our RBC has nucleus, isn't it? Oh, he's telling yes. Okay. So what happened? But the progenitor cells are having nucleus. So what happened to this nucleus? They are removed from the cells. So that is another observation. Also, the nucleated cells has different organelles like mitochondria, like Golgi bodies, like endoplasmic reticulum. We will see, are they present in the cytoplasm of RBC? No. So they will also be removed. Isn't it? So nucleus will be removed, cytoplasm will be removed, and the removal of nucleus will see certain changes. The removal of RBC is called Pyknosis. 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 What is Pyknosis? You may be asked. Okay. So by the process of Pyknosis, nucleus is removed. And one more thing, the progenitor cells, hello, listen, the progenitor cells are not having any hemoglobin. But RBCs are having hemoglobin. So that means in between, hemoglobin is added, loaded in the cell. Isn't it? So that is another change. That is another change we will be noting. Isn't it? So in this way, another change. What is the change? Matured RBC is disc-shaped, biconcave, but the progenitor cells are not like that. They are round. They are granular shape. That means there will be shape changes. So now tell me, what are the changes you will be observing? Number one, number of cell will be increased. Number of hematopoietic, this progenitor cell, if it is one, then RBC from this progenitor cell, lot of RBCs, so their number is increased. Secondly, by mitotic division. Initially, mitotic division is fast and then gradually fall. Okay, and ultimately it will be stopped. This is number 2 observation. And number 3 observation, nucleus will be removed or organelles will be removed. Okay, so that is another observation. Hemoglobin will be added and there will be change in shape. Okay, fine. If you can write these points, no, a lot of marks. Okay, and different stages. Okay, and the factors involved in different stages. Okay. When you are in your mother's womb and your age was just 1 week, 5-7 days, 1 week, 2 weeks, 3 weeks, at that time your blood cell formation started. At the end of 7 weeks or during 7 weeks, your heart started to develop, not only started beating. When? During 7 weeks. So if you study any fetus at 7 weeks, you will get the heart is little bit and their heart is beating like that. Okay. So, and see, heart is beating, what is the function? Pumping blood, definitely blood is produced. So at that time, who are producing blood cells? It is the mesoderm. Ectoderm, endoderm, mesoderm. It is the mesoderm, and from this mesoderm's blood vessels are produced, and this mesoderm of yolk sac, they produce blood cells during first 3 months. During first 3 months, what is the site? Yolk sac. And after next 3 months, what is the site? Site is changed to liver and spleen. To liver in the next 3 months. Second trimester, we call trimester. In second trimester, they are formed in liver and spleen. And after that, last trimester, and bones are developed. All bones will produce RBCs. And after birth, these bones, only these bones will be producing And during this is the developmental stage in childhood, and the bone has red bone marrow, yellow bone marrow. So initially all were red bone marrow. Now gradually yellow marrow starts developing, occupying the position, and the red bone marrow pushed towards in the long bones. So transformation of yellow, the area that is transformed into yellow marrow, they are not capable of producing RBC. Only red bone marrow can produce RBCs. So in adulthood, what? Not all bones, only the membranous bones, only the membranous bone can produce erythrocytes. Okay? What are these membranous bones? Vertebra, sternum, ribs, skull bones, pelvic bones, proximal ends of femur and humerus. Humerus, femur. Okay? So this, any doubt regarding site of production of RBC? Any doubt? Can you remember all the points? Huh? Yolk sac. Mesoderm of yolk sac. First 3 months, yolk sac. Next 3 months, liver and spleen. And last, all bones. And during childhood, all bones. And gradually, the membranous bones, it will be limited only to membranous bones. Clear? Okay. Now, see, it is the period of months, gestational months, first week, second week, third week, this way, and it is the age after birth. And what are these? See, what is this? Liver. Liver start producing from the second week and stops here. And the yolk sac stops here. And this spleen starts here, stops here. And bone marrow, rapidly, in childhood, all the bones produce. And thereafter, all bones cannot produce, only the membranous bones can produce. Okay, so this is informative. Now, the stem cells, what are the stem cells I told? From the stem cells, we will get what? The stem cells that are that from the blood cells that will be produced from one kind of stem cell. What is this? Hemopoietic stem cells. Stem cells differentiated into hemopoietic stem cells. Now these hemopoietic stem cells, okay, they are capable of producing all type of blood cells, and they differentiate into one or any other type of committed stem cells. Committed stem cells means you are committed to produce only RBC. You are committed to produce only megakaryocyte. You are committed to granulocyte. Okay, so this sort of committed stem cells will be produced. Is that clear? Okay, fine. Now you can see this pluripotent PEAs, pluripotents. Pluripotent means, as I said, they can produce any type of cells. So pluripotent, hemopoietic, but these pluripotent cells are only limited to hemopoietic cells. They cannot produce liver cells. They cannot produce muscle. Okay, so they can produce only the blood cells. Now it will be dividing. They will be dividing into this. What is this? Colony forming unit, spleen, when it is in embryonic stage. And it will be divided into colony forming unit blast cells. Burst. These are means, whenever you will find, they are capable of dividing. They can divide. Okay. But when you find a site, that means it has stopped. Okay. So from this we will get CFU, colony forming colony. When these cells are kept in a medium, all factors are given. for development, then will be a lot of RBCs are generated. That means they form colony of erythrocytes. So from this colony forming erythrocytes is said, and they by different stages produce erythrocytes. This is not a single step. This is not a single step. This is not a single step. Multiple steps are involved. Let's see. Sequence. Okay? So from PSSC to mature RBC, how much time is required? 21 days. And what is the progenitor cells? This is the progenitor cell, pro-erythroblast. From here to here, it takes 7 days. Okay? Out of these, 2 days are required from here to here. And one important thing you will observe, this cell, pro-erythroblast, its diameter is 20 micron. How much? 20 micron. And they will gradually decrease in size, because we know the size of mature RBC is 7.2 micron. So what happens? Gradually their size is decreased. Okay? See, in the first step of pro-erythroblast to basophilic, one T is absent, blast, T is there. Erythroblast. Basophil means why the name has been given? Because the cells has affinity for basic dyes. As a result, the cytoplasm of these cells can be stained with basic dye and they take blue coloration. Okay, so cytoplasm is blue. Why blue? Because they take basic dyes. Okay, that's why their name is given basophilic. Okay, basophilic erythroblast. This will also be having. And here, polychromatic. Try to understand the meaning. From this stage, hemoglobin starts appearing, giving a color to the cytoplasm. That's why chrome, red coloration comes. If you stain it, you see, it will take pink color. Okay, so these dyes are acidophilic. And so there are the cytoplasm is mixed. Some will be basophilic, some because hemoglobin is added, so it will give red coloration. So it is polychromatic. That's why it is called polychromatic, multiple colored granules. So polychromatic, the term polychromatic means it. After that, orthochromatic. Okay, again the cytoplasm will be alkaline. Okay, then the mature RBC is acidophilic. Okay, so in this way, the color of cytoplasm, reaction of cytoplasm also gradually changes. Isn't it? So that is also another important characteristic changes during erythropoiesis. So what are the steps? Pro-erythroblast means then it will develop blue color cytoplasm, that is basophilic. Then it is polychromatic, both will be present. That's why it is polychromatic. Then it is again it will be changed, orthochromatic. Then reticulocyte will be formed. Then mature will be formed. Can you try to remember all the sequences? Try to remember all the sequences. Now what happens? As I said, changes during erythropoiesis. Take this photo. Try to remember all this point. Taken? Okay, fine. Now the stages. And what are the external factors are involved? The external factors that are involved are some hormones. What are these hormones? Two important hormones are involved. And these are interleukin. What? Interleukin. abbreviated as IL. These are formed from T cells. Did you hear the name T cells? What are T cells? Lymphocytes, our lymphocyte, we have studied blood cell. One kind of blood cell is lymphocytes. Lymphocytes are of three kinds. What are the three kinds? They transform into three kind of, they develop three kind of, one is B cell. And B cells provide immunity. And what kind of immunity? B cells? Humoral immunity. They will produce antibodies. Another type of is T cells. They provide cellular immunity. Okay, they also fight with antigens, foreign antigen, pathogens. The mode of action is different. And another is, who said? Good, study, very good, excellent. So, keep on encouraging me like this. Okay, I feel encouraged. Okay, so you try to respond. All of you, try to respond. Don't feel shy. Okay, and this killer cells function, killer, the invaders, kill him. They are the killer. There, I will tell the mechanism, all this mechanism I will explain in our future classes, in immunity classes. So, these T cells, they produce cytokines. What? T cells produce cytokines. T cells produce cytokines. And these cytokines are classified. They produce, these cytokines are nothing but IL, interleukins. And these interleukins are of different types. IL-1, here, which interleukins will be involved? Many types of interleukins are there. Here only IL-1, IL-6 and IL-3 will be involved. They will stimulate. They will give stimulation. Hey, divide first. As a result, what will happen? These stem cells, hemopoietic stem cells rapidly will form burst-forming unit, erythrocytes. And whenever it is produced, they will produce CFU, colony-forming unit, erythrocyte. Okay, if you don't have, if your body cannot produce sufficient lymphocytes, what will happen? Do you think there will be proper erythropoiesis? No. If your body doesn't produce sufficient amount of erythropoietin, or in short we abbreviate with EP, then what will happen? If no EP is there, EP helps here also. This erythropoietin also here, acts here also. Erythropoietin also acts here. Basophilic, polychromatic, yes, here also. Erythropoietin acts. If you don't have erythropoietin, there will be less production of RBC, very important thing. Okay. One patient came to you and he said, I'm very sick, I can't walk, I feel always drowsy, sleepy. That is, he is suffering from hypoxia. He is less energetic. Hypoxia. Why hypoxia? Less test blood. Why? Hemoglobin is less. Why hemoglobin is less? Why hemoglobin is? You have to study whether there is problem in bone marrow or in different steps some globins in the lot of things are required. Whether all, for example, iron, for example, B12, for example, folic acid, zinc, they are provided sufficiently. You observe, they are taking this. Then what is the problem? Then the patient said, sir, few days back, I had to remove one of my kidney. And because of this, the patient developed anemia. Removal of kidney developed anemia. Removal of stomach part because of cancer. Removal of part of the stomach developed anemia. Arey bhai, what's the reason? How kidney is related to bone marrow? Because 75% of the erythropoietin is synthesized in the kidney. No kidney, both kidney, lot of erythropoietin, one kidney, less erythropoietin, and a patient suffering from chronic kidney disease. Kidney is gradually shrinking. It happens in diabetic patients. Chronic renal disease. In this, renal tissues are going, atrophied. So that means the cells that are producing erythropoietin, they are also going. As a result, there will be lack of erythropoietin. So less erythropoietin means this stage. What will happen to this stage? Less conversion. What will happen to this? Less conversion. What will happen to this? Less conversion. So it will affect you. Sir, erythropoietin. Kidney doesn't produce erythropoietin. Stomach doesn't secrete erythropoietin. Then what is the relation? Okay, think it. When I will be describing the factors, one of the factors is vitamin B12. If vitamin, your part of the stomach is removed, then what will happen? Because of absence of this, vitamin B12 will not be absorbed properly. Why? The stomach, the parietal cell that produce hydrochloric acid, it has got another name, oxyntic cells. Parietal cell or oxyntic cell. What is their function? To secrete hydrochloric acid. We know hydrochloric acid needed for our digestion. Or other function, activation of enzymes, and with the food, lot of foreign bodies are coming to kill them. So hydrochloric acid, lot of function. That cell also produced another thing. It is called intrinsic factor. What? Intrinsic, very important. When stomach is removed part, we don't get intrinsic factor sufficient amount. But this is required for carrying of vitamin B12 from stomach to the large intestine, ileum. Without this intrinsic factor, they cannot be taken to the ileum. And ileum is the portion from where vitamin B12 is absorbed. So no intrinsic factor, no absorption. No absorption of B12. I know vitamin B12 is necessary for DNA synthesis. Folic acid is necessary for DNA synthesis. Remember this point. DNA synthesis. Can you think without DNA, cell multiplication? Chromosomes has to be divided. Numbers has to be double. So chromosomes, what is there in DNA? DNA strands are there. If DNAs are not produced, then how the cell will divide? How this will go on? That's why vitamin B12 is essential. Deficiency of vitamin B12, it will lead to anemia, and this has got a particular name, right? Pernicious anemia. What is this? Pernicious anemia. I'll come, Pernicious, right? Pernicious anemia. Okay. Similarly, another factor is involved. There is folic acid. Okay, I'm finishing it first. Time is running out. So these are the different steps. Have you written this? Okay. From pro-erythroblast to mature RBC, how the size gradually coming down. Here it was 15 to 20 micron. This cell, progenitor cells. Then the size is reduced to 14 to 17 micron. And then here where the RBC hemoglobin starts appearing, that's why polychromacia means polychromacia means chroma. So, these are coming to appear because of hemoglobin started appear. Previously it was basophilic, so now it is acidophilic. So that's why it is giving polychromacia. It is the step. You have to remember from in which step polychromacia is seen, in which step hemoglobin starts appearing. Okay, Bible question. Okay, then what happens here? During this phase, gradually the size is decreasing. Another thing you will be noting in the nucleus. Initially the nucleus there is 2 or 6 nuclei. Nucleolus. As you proceed, the nucleolus will be going out. Then the nucleus will be condensed. They will be condensed gradually. Okay. After condensation, they will take a form of a ink dot. Ink dot. They will take the formation of a ink dot. Okay. Now this ink dot means the nucleus will be coming out of the cell. And what is the process? Pyknosis. And this ink dot is called Pyknosis. Pyknotic nucleus, it is eliminated. Ejection of nucleus. So these are the various changes you will be noticing in different states. You have to write. Okay. Normal nucleus, when it is transformed into condensed into like this, and then it is fragmented, then eliminated. And this fragmentation of nucleus is called Karyorrhexis. Karyorrhexis. Then what is this karyolysis? It has gone out. Lysis. All the components are lysed here. Okay. These efferents of in this way, either they will be thrown out, either they will be disintegrated in the cells. So all this I have explained. Reticulocytes, the reticulum. Why the name is given? Reticulum. What is this reticulum? Reticulum is the cell organelles. They were not thrown out. They were not thrown out of the cytoplasm. They were within the cytoplasm but destroyed. Okay, and this destroyed part of the organelles, they produce chromatin. So, presence of chromatin means it is the debris that form reticular structures. Okay, so in the next step, the step in which we will observe this, that is, it will be termed as reticulocyte. And in mature RBC, it is absent. Everything is removed. So, this mature RBC will be formed. Okay. What is reticulocytosis? When there is increased activity in bone marrow, demand for RBC is much more, then immature reticulocyte comes into circulation. As a result, their number is increased. This is called reticulocytosis. If one patient came to you, he is having Pernicious anemia. Okay, then you started treating. Whether the patient is responding, how will you know? If you find this will be formed in the bone marrow. If you find in the blood there is more reticulocytes are coming, that means your bone marrow is the patient's bone marrow is active. Means it is giving result. Okay. Reticulocyte has no nucleus, has no organelles, is larger than the mature RBC, is not concave, is not biconcave, has many polyribosomes. In severe anemia, many of are released into the blood prematurely. It is called reticulocyte response. And the process is reticulocytosis. Already told the durations. These are the changes you notice. Reticulocytes, I already told you. Now the factors. The factors will be dividing into three main classes. Only 15 minutes, I have to first cover it first. What are these? One are hormonal factors, then the dietary factors, then there is other factors. Among the hormonal factors, most important, as I said, is erythropoietin. Erythropoietin. Where from they are secreted? Mainly in the kidney. They are also formed to some extent in the liver, also very less from the brain. And evidence has proved that uterus can produce to some extent, very little. But these are not significant. Okay, so significant is kidney. Now, kidney is having a lot of structures. From where? From peritubular. What is peritubular? You think. Bowman capsule, proximal convoluted tubule, loop of Henle, then distal convoluted tubule, and they are taking so many important things, sodium ions, everything are there. So they may be eliminated. So here, this distal convoluted tubule are encircled or side by side, there are blood vessels, capillaries, they are absorbing, they are secreting something, they are absorbing. These are peritubular capillaries. And these peritubular capillaries, you understood this? Okay, so erythropoietins are synthesized from where? From the peritubular capillary endothelial cells. And these endothelial cells are very sensitive. Very sensitive to what? To hypoxia. If blood is having less oxygen, less oxyhemoglobin, they can sense. They are sensor. What is happening? In blood there is less oxygen. So I have to produce erythropoietin. So they immediately secrete erythropoietin in the blood. This blood goes to bone marrow, starts dividing cells, starts erythropoiesis. Isn't that interesting? So that is the function of erythropoietin. It is glycoprotein in nature. Means glycoprotein means glyco means carbohydrate. Glyco means carbohydrate and protein. These are conjugated protein. And this glycoprotein, this part, glyco part, carbohydrate part is having, this can bind with erythropoietin receptors that are present on the target cells. So they are the target cells, which are the stem cells are target cells. Erythropoietin acts there. So they must be having receptors. So this glycoprotein, the carbohydrate part actually binds with this receptor of the target cell. Then erythropoietin starts its action on the cell. Means it will go, okay, start dividing. Some changes will be made within the cell. Within the cells, where this will cause rapid division of the cell, rapid mitosis of the cell. So in that way, that is the mode of action of the erythropoietin. Study G.K. Pal. Which book? For this. Okay. For erythropoiesis and factors affecting G.K. Pal. And you can also study this Shambulingam also. So first go through the Shambul, it is give easy explanation, then enter into G.K. Pal. Okay. So erythropoietin, then androgens. Male are having more blood cells than female. Why? Because of androgens. They are having testosterone. But before puberty, all are having same amount of, no less, no more. But after puberty, they have started secreting more testosterone, androgen, and that stimulate. And estrogen is having opposite effect. It cannot increase, but it can decrease. And you see, while cells divide, do they require more energy or less energy? More energy. My activity is more. And one hormone is required for producing more energy. What is this hormone? If this hormone is increased in your body becomes like, whenever somebody touches, your skin temperature is high. No? And when its level is less, feeling cold. Though it is not the winter season, feeling cold. Oh, switch off the fan, I'm feeling cold. Hypothyroidism. And when there is hyperthyroidism, there is increased temperature, production of temperature in the body. Heart go on. Palm, and this area becomes very hot. Okay, so hyperthyroidism means increased temperature means what? Increased production of increased metabolism. So here the cells also need increased metabolism. So that's why thyroxine helps here the production of more ATP, conversion of carbohydrate to form more ATP. That's why thyroxine stimulates erythropoiesis. Got it? Similarly, that boy, hello. Adrenaline also increases erythropoiesis. Okay? Estrogen, thyroxine, anti-pituitary growth hormone. The way growth hormone divides your body, makes growth by increasing the cell number, the similar way erythropoietin also acts. So growth hormone will also increase erythropoiesis, isn't it? So try to relate that way. Then you will be able to answer. Similarly, some hormone, TSS, because it is controlling the thyroid gland, and thyroid gland is, thyroxine is providing energy to the cells, dividing cells. So that way it is correlated. ACTAs, they form, it is because of this, in in adrenal gland, cortisol is produced. Cortisol is produced, and cortisol increases erythropoiesis. Interleukins, IL-135, we have seen, they are involved. In particular the addition. Now the dietary factors. Okay. You are having less hemoglobin, eat fish, eat egg, eat milk, eat meat, chicken. Why? You require more protein. Proteins are of two classes. One is first class, another is second class. First class proteins are having all the essential amino acids and in sufficient amount. But second class proteins are having less essential amino acids. We require, actually our body can't prepare these essential amino acids. That's why we depend on murgi, egg, anda. We depend, they provide us first class protein. Milk, they provide us first class, because all these first class protein are all supplies all the essential amino acids. Try to remember in biochemistry all this. Name of this essential amino acids. Glycine is one of them. Methionine is one of them. Lysine, you will find in the tonic. You will be writing to the patient, sir, give me one tonic. It is mental tonic. Otherwise, it is not going to help. Lysine is there. One of the essential amino acids. Okay. So, in the protein is very much. Otherwise, hemoglobin, globin, for globin synthesis, amino acids required. Who will provide amino acids? They say exogenous protein. So that's why it is needed. And minerals, without iron it is not possible. So we have to provide minerals. And copper and cobalt is essential for utilization of iron into the hemoglobin. They are supporting incorporation of iron into the cell. That's why copper and cobalt, nickel, these are essential. And cobalt is one of the component of a vitamin. Name them. Name this vitamin. Cobalt is the constituent of a vitamin. Can you name the chemical name of vitamin B12? Cyanocobalamin. Cobalamin, that is, it is having cobalt. No cobalt, no vitamin B12, no vitamin B12. This is 11. See how beautifully they are related. So every time ask questions, why it is? Then you will get the answer. Then you will be remembering this. Okay. And other factors, intrinsic factor. As I already told, your stomach produces one intrinsic factor. Without this, no vitamin 12 will be absorbed from the ileum. So you will suffer pernicious anemia. Okay. Then chemical factors are there. Some drugs may be involved, and environmental factors. Hypoxia, low blood, low oxygen pressure, as in high altitude. Okay. Any condition that is not preventing you to take less oxygen. One is emphysema, asthma. In such condition, there will be hypoxia. This hypoxia will stimulate. So in them, to counter this situation, they produce more and more RBCs. Okay, and environmental factors. So I think I covered everything. So if you can learn these points, okay, in your question, term and exam, this question from here most. So you may be Cassell's hemopoietic principle. What is this? Hemopoietic principle is stomach, the vitamin B12, folic acid, and the intrinsic factor. They together form Cassell's hemopoietic principle. Okay, vitamin B12, folic acid. Folic acid, actually, folic acid is also needed for DNA biosynthesis. So, if no supply of folic acid, then there will be anemia. And you can identify, this is because of folic acid. How? Macrocytic RBC. Megaloblastic RBC. So, see, megaloblast means in the stage, these are megaloblast only. These megaloblast are releasing to blood in such anemia. Will there will be presence of hemoglobin? These cells started appearing in blood, so they haven't gone through these changes. Hemoglobin is not added, so less hemoglobin will be there. Big size RBC but less hemoglobin. What is this condition? Macrocytic hypochromic. Isn't it? So these sort of things you have to remember. Fine.